Hi everyone, today's topic is tumor suppressor genes. A tumor suppressor gene TSG or anti-oncogene is a gene that regulates a cell during cell division and replication. If the cell grows uncontrollably, it will result in cancer. When a tumor suppressor gene is mutated, it results in a loss or reduction in its function. In combination with other genetic mutations, this could allow the cell to grow abnormally. The loss of function for these genes may be even more significant in the development of human cancers compared to the activation of oncogenes. Tumor suppressor genes can be grouped into the following categories: caretaker genes, gatekeeper genes, and more recently, landscaper genes. Caretaker genes ensure stability of the genome via DNA repair and subsequently, where mutated allow mutations to accumulate. Meanwhile, gatekeeper genes directly regulate cell growth by either inhibiting cell cycle progression or inducing apoptosis. Lastly, landscaper genes regulate growth by contributing to the surrounding environment. When mutated, can cause an environment that promotes unregulated proliferation. Next examples: RB, the retinoblastoma gene. Retinoblastoma is a cancerous tumor of the retina. Pathway of RB: diverse mitogenic and oncogenic signals induce the expression of D-type cyclins that activate CDK4 and 6. The resultant kinase activity is balanced by the CDK4 and 6 inhibitor P16 Inc4A. Typically, a P16 Inc4A is at low levels in cells but can be induced by oncogenic or the DNA damage stresses to suppress CDK4 and 6 activity. These signals coalesce or regulate the phosphorylation of the retinoblastoma tumor suppressor. When active, unphosphorylated or hypophosphorylated, the RB represses the activity of the E2F family of transcription factors and limits the expression of a program of genes required for S phase. For example, MCM7 and CDC6, and G2 by M progression. For example, CDK1 and cyclin B. Phosphorylation relieves this transcriptional repression and allows for cell cycle progression. CDK, the cyclin-dependent kinase, P phosphorylated. The cancer of the retina occurs in two forms: familial retinoblastoma and sporadic retinoblastoma. Sporadic retinoblastoma. पहले familial. Multiple tumors in the retinas of both eyes occurring in the first weeks of infancy. Sporadic retinoblastoma. A single tumor appears in one eye, sometimes in early childhood before the retina is fully developed and mitosis in it ceases. नेक्स्ट फेमिलियल रेटिनोब्लास्टोमा, इनकी डिटेल देखें. Familial retinoblastoma occurs when a baby inherits from one of its parents a chromosome number 13 and has its RB locus deleted or otherwise mutated. The normal RB protein controls the cell cycle. It integrates the signals reaching the cell to determine whether it is safe for cell to complete the passage from G1 of the cell cycle to mitosis. Next mechanism. The unphosphorylated RB protein prevents cells from entering S phase of the cell cycle. It does this by binding to transcription factors called E2F. It does this by binding to transcription factor. This prevents the E2F from binding to the promoters of such proto-oncogenes as c-MYC and c-FOS. Transcription of c-MYC and c-FOS is needed for the mitosis, so blocking the transcription factors needed to turn on these genes prevents cell division. However, if conditions are adequate for the cell to successfully complete mitosis, the RB protein becomes phosphorylated, releases the E2F, and the cell can proceed through the cell cycle.